All right, so we'll look at that. I will see possibility of doing it as fast as possible. Now, cytology means study of cells. Physiology means the cell function. And obviously, your cell is the structural and functional unit of life. Structural means that's the smallest structure that have the function of life. That's the smallest structure that can be called living organism or living thing. Functionally, functional unit also means is the smallest unit that have the function of life, that can perform the functions of living thing. And this is your cell. Let's go. Now the cell there are major structures that are evidently in all cells is the plasma membrane. The plasma membrane is the same thing as cell membrane, and also the cytoplasm, then a nucleus. Now, in this cell, for instance, now, this is a plasma membrane. Here is the plasma membrane that surrounds the cell. You see, that's the plasma membrane. Now, the cytoplasm is everywhere here. Everywhere here is cytoplasm. Everywhere here is cytoplasm. And here is the nucleus. Now, this is the nucleus here. This. Now, usually this nucleus is at the center of the cell. But there are some cells that doesn't have nucleus. And I will be talking about that in a bit. So usually every cell have the plasma membrane, the cell membrane, the cytoplasm, which is what I just showed you. Inside the cytoplasm is cytosol. The cytosol is liquid. All right? It's like semi-liquid. All right? The cytosol is a semi-liquid. And think about the cytosol like a gel inside the space. Cytoplasm is a space. All right? Cytoplasm is space, is not a structure. All right? Cytosol is semi-fluid or semi-liquid. So cytoplasm is space. Cytosol is liquid. It is in this semi-liquid that organelles are embedded in, which is what you see here. That's what you see here. These are organelles, right? These are organelles. All these are organelles. Organelles, 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 organelles. These are all organelles inside the cytoplasm. And the fluid that is inside the cytoplasm is the cytosol, whereas the space is the cytoplasm. This is the nucleus. Now, if a cell does not have nucleus, what does it have? It has DNA. And the DNA, the location where the DNA is, we call it the nuclear region, but I'll get there. Let's go. Now, when you look at the cytoplasm, I mean the plasma membrane, for instance, it's made up of two structures or two substances. Majorly, it's made up of phospholipid, phosphate and lipid. Now, you remember phosphate that we have in nucleic acid, right? So these are made up of phosphate and lipid. Now, so the cell membrane proper is actually this. It's like this: phosphate edge and fatty acid tails. The fatty acid are the lipid. So this guy is the one that is hydrophilic. Okay, I can do this. So these guys are the hydrophilic, right? You have it there, rather. The hydrophobic are the fatty acid. Now, any substance that is hydrophilic and hydrophobic, or that have both the hydrophobic and hydrophilic part, we call that substance amphipathic molecule. Anything, any structure that have both lipid and hydrophobic part, both hydrophobic and hydrophilic part, we call that amphipathic structures. All right? So the cell membrane is amphipathic structure, meaning that it has both layers, both hydrophilic and hydrophobic part of it. Now, it's arranged in two layers, so we call it bilayer, phospholipid bilayer. And if you look at this, it will show you what I'm talking about. So this is a cell membrane, for instance, and you can see that it has two layers. Two layers are there. These are the two layers. This is one layer. This is another layer. So there are two layers of membrane that are facing each other, inverted on each other. And the reason you have this is so that if you see this side is hydrophilic, the fatty acid side is hydrophobic, this side is hydrophilic. The purpose of having double membrane is not to have a larger thickness, no. The purpose of having the double membrane is simply for adaptation. It's simply for adaptation. So, for instance, now you can say that the cell membrane, the hydrophobic philic part is what you see on the extracellular space, because this space is going to be filled with water too. There's going to be water here. So if there's going to be water here, then the part that will be in contact with this must have water. And this part as well will have water inside the cytoplasm. So that is why it is inverting on each other to make two layers so that both sides are water-friendly environment. Now, if you look at the cell membrane, you can see proteins here. These are proteins. These are proteins. These are proteins embedded in the cell membrane. These are proteins embedded in the cell membrane, and you can see cholesterol there. You can see cholesterol there. Now, what we call fluid mosaic model is when you have structures like protein in the phospholipid bilayer, and because they are embedded in the lipid environment, they are flow thin. So they are able to move around, and that is what we call fluid mosaic. So it's like fluid mosaic, proteins embedded in the membrane that is fluidy, that is able to allow movement. All right, let's go. Now, the proteins on the cell membrane, what are their functions? Now, there are some proteins on the cell membrane that are passing through the entire cell membrane. Those kind of protein, we call them integral protein. You can see, like this kind of protein now is going through the entire membrane. You can see it pass through the entire of the membrane, the entirety of the membrane. That is an integral protein. If you see like this now, it goes through the whole of the membrane. It's integral protein. Both, when you have proteins, for instance, that are just on the surface. So let's say you have a protein now that is just like, you can only see the protein here. That will be peripheral protein. And so if you look at this one, this protein, this one is peripheral protein because it's just on the surface. It's not going through the entire length. It's not going through the entire length of the membrane like all these other ones. So those are peripheral proteins. Like this now is integral. It's integrating the membrane. But proteins that are just on the surfaces. So if you have a protein here, for instance, now, that will be surface or peripheral protein. All right, let's go. Now, what are the functions of this protein that are inside the membrane? Number one function they do is they could be for cell recognition. All right, they help cells to identify each other. Now, some of them, they also help to attach a cell to another cell. Now, you know that tissues are a group of cells that work together to perform a common function. That is what tissues are. Now, if that's tissue, how do they come together to perform the function? They have to be connected together. And for them to be connected together, there has to be something connecting them. One of those things is protein are the one that connects them. They join the cells together so that the cell can work together as a tissue. All right? There's a protein that we call connexin. This connexin now, it's a kind of protein, and it helps to join cells together. Connexin. All right? They help to join cells together to form tissue. It's a kind of, we call it gap. it's a kind of gap junction rather. So we call it gap junction. It's connecting, it makes a tunnel, we call it connection, and that allows molecules to pass through the cell. But it's all I'm saying is that connects 2 cells together, 3 cells together to form a tissue. Now there are some other proteins that help to speed up chemical reaction. Those are enzymes. Most enzymes are mostly proteins, right? Then we have some proteins that are called receptor. What the receptor does is, if an hormone is released into the bloodstream, there will be a receptor that will accept the hormone. So the receptor will be on the membrane, just like what you have here. These are the receptor guys here, and then they are going to allow either hormone or ligand or anything, substance that needs to bind on them. If the receptors are not there, then those substances will not be able to bind. Those hormones will not be able to perform their functions if the receptors are not there. Function we have here is for transportation, which is key. All right. Now, let's say this is like glucose now, for instance. Glucose is a large molecule. It cannot diffuse through the cell membrane. It needs to be carried by a protein, which we call glucose transporter. Now, when glucose transporter pick a glucose, then you can take it into the cell. But without that, the molecule glucose cannot get into the cell because it's a large molecule, right? Okay, let's go. Now, plasma membrane. Your plasma membrane is selectively permeable. Selective means that it does not allow everything. Permeable means access, entrance. So the plasma membrane is selective to what it gives access to. It does not give access to everything in your body. It gives access to something. It does not allow some things. And that's what I just talked about. For instance, now glucose, for instance, cannot pass through the cell membrane by itself. It needs a transporter. So some chemicals can pass through the cell membrane, whereas some chemicals will require a transport protein for them to be able to pass through. The cell membrane is made up of lipid bilayer. I've told about it. It's mostly hydrophobic, permeable to small, non-polar and uncharged molecules. So think about it. Permeable to small molecules can pass through the cell membrane, can diffuse. Non-polar molecules can also, then uncharged molecules can. Uncharged molecules simply means neutral molecules or neutral atoms. Now, the cell membrane will not allow larger molecules like glucose. It will not allow polar substances, and it will not allow charged molecules, right? Like ions, NH to Na plus, potassium plus, sodium chloride, chloride, like Cl minus, all of those will not pass through the cell membrane by themselves. Now, the cell membrane is permeable to water, but not it's a restricted pace, because water is also a polar substance, right? But this water can diffuse, but in a very limited quantity. So there's also a protein that helps to allow water transport, and that is called aquaporins, right? Integral protein, they have as channel or transporter, they help molecules that cannot pass through the membrane. Let's go.
I have a question.
No. Go ahead, please.
Sorry to interrupt, class. So my question is, I believe on the other slide it said that uncharged atoms cannot pass through the membrane, and I'm wondering if that has to do anything with the anions and the cations. So
Yes.
a cation is a positive, and a negative is a anion. And I'm wondering if is it the anions that you're talking about when they're unable to go through the molecule?
You see that? You see what I'm writing?
Yes.
So when you say charged molecules means ions. It means anything that have charges. So let's say you have this now, so ions are cation on anion. Cation is positive, right? Anion is negative, right? So something like Na plus now cannot go through the cell membrane by itself. Does that make sense?
Yes, sir.
It will need a transporter. Something like potassium ion cannot pass through the cell membrane by itself. So something like this now, SO42 minus, it cannot pass through the cell membrane by itself. Anything that have charges, and when we say charges, we mean positive or negative. Does that make sense?
Okay, so that includes both positive and negative.
Exactly, it's both positive and negative. Anything that has positive or negative cannot pass through the cell by itself. It will need a carrier. It will need help. They don't diffuse through the membrane. Did I answer your question?
Yes, sir. Thank you so much.
Now, it says charge. It's not just positively charged. It's not just negatively charged. All charges. Thank you. Now, you are not interrupting me if you are asking a question. That is why it's not just a recording. That is a live lecture, right? So I want you to ask questions when you have questions. That's what makes it interesting to me as well. So feel free to ask. You are not interrupting. You are not disturbing. You are simply helping me to do my job.
Thank you. And so we won't have to worry about active transport or passive transport or anyway, because this is a A&P class, correct?
We'll worry about that a little.
Okay, thank
you. Just a little bit. The reason we are worrying about it is because we are looking at the cell, and for cell, when you understand the cell is also, we look at, because it's A and P too, that's why we have to look at it. And because it's anatomy and physiology, the structure and how they function. So now what we are looking at here now, even though it may look little, but it's quite significant, because what we are looking at here is that the structure of the membrane determines the kind of function it allows. So anatomy, structure of the membrane, physiology, function that it allows. Does that make sense?
Yes, sir.
Good. Thank you. So now, so active transport and passive transport. Now, what's the difference between active and passive? Passive is just, when you say somebody is passive, what does it mean? I say this person is just passive.
Lower energy, easygoing.
Okay. Go ahead. Anything else? Lower energy, easygoing. Anything else? When you say this person is just passive. Now, being passive, no response, okay? Now, we can also, there's nothing we have said that is wrong. I'm just looking for ways to connect it to what we are studying, okay? Now, when you say something is passive, you are saying that things are it's just happening, like, it's just happening. It's a car that has no driver. It's just allowing everything to just happen. Things are just happening by themselves. No special input, nothing. It's just a passive process. I'm just watching what is happening. It's just passive. All right. Now, a passive transport is a kind of transportation that does not require energy. Now, it does not require energy at all. So if I have this mouse now and I want to drop it, I don't need energy to drop it. The reason I'm not needing energy is because it's going from a high location to a lower location. So when a chemical is going from high concentration to lower concentration, in general, it does not require energy. At any point, when a reaction is going from where it is high to where it is low, it does not require energy. It only requires energy when it's going from low to high. But if it's going from high to low, no energy is required. So that's why we call it passive. If it is active, it means it requires energy. And that will be the case when it's going from lower to high. Now let's go and see it. Now, this is passive transport, simple diffusion. And simple diffusion simply means that a chemical is moving from where it is high, concentration is high, to where it is low. At such point, it does not require energy to occur because it is going from where it is high to where it is low. Now, no energy is required. That's why we call it passive. No protein is even required. That's why it is passive. Now, example will be movement of water, movement of ions, movement of oxygen, and movement of carbon dioxide. And a typical example of what we are talking about is this. Look at this chart here. If you put sugar, for instance, in this water molecule, now you know we had said large molecules does not diffuse, does not pass through the membrane, but I want to illustrate this here. So look at what I'm showing you here. If you put sugar in this water now, you are going to just put the sugar there and then you leave. You don't necessarily need to mix it before the sugar will circulate and make up the whole thing. If you put a drop of dye inside water, it will circulate by itself. You don't need to move it. You don't need to do anything. If you put orange juice inside some water, it's going to circulate and everything will turn to the color of the juice in a bit. That is diffusion. The same thing when you spray a cologne in a room. People in the room will start to, if you just spray it now, people in the room will start to perceive it without you doing anything. You don't need to be active about it. It's just a passive movement, all right? So that is what we call passive transportation, passive movement. So, and again, we've said it, molecules, smaller substances, substance that does not have ions, they can pass through the cell membrane using diffusion. The other kind of transportation is what we call osmosis. Now, see the difference between osmosis. If you want to summarize it, osmosis is simply the diffusion of water, or change it to the diffusion of solvents. That is osmosis. It's simply diffusion of solvents. Now, what that means is that what is moving here to maintain balance is water molecule. Here, diffusion, what is moving around here is the solute, all right? Or the solute or other molecules. But in osmosis is the solvent or water molecule that is moving. So the water moves from where there is more water to where there is less water. This thing is as simple as this. Let me bring you this illustration here. Look at this. In this environment, this is isotonic environment. Isotonic environment for us, think about this as a red blood cell, is when you have 0.9% of NaCl. So 0.9% salt solution is our isotonic environment. So water, this is water, 0%. And solute is going to be, let's say 10% NaCl. So you put your cell, I'm painting different scenario now. If you put your red blood cell in this water, in this solution, now the isotonic environment for us, and you know that when you go to hospital and they need to give you normal saline, if they want to give you, they want to put fluid into you, what they are giving most of the time is first normal saline, which is 0.9 or 0.8 something % sodium chloride. Now, if you put red blood cell in that environment, that is okay. The red blood cell is going to be fine in that environment. But if you put red blood cell in a water environment that is 0% NaCl, what is going to happen? Or 0.1 NaCl, what is going to happen? the water that is in the environment will want to come, because this environment means there's a lot of water and there's less water in the cell. So the water is going to move into the cell. And when water moves into the cell is going to swell and then burst. This kind of process, when the cell bursts like that, we call it hemolysis. Now that is animal cell. Then, if you put the same cell in a solution that has too much quantity of salt, we say that solution is hypertonic. What is going to happen? There's too much salt here. And you put your red blood cell there. The salt solution will take water out of the cell because the environment needs water. And now this cell is going to shrink. When the cell shrinks, we say the cell is crenated, crenation. This is actually the same thing. Think about it. If you see an insect or an ant and you put salt on it, you have invariably killed that ant because it's going to die, because the salt will dehydrate the ant. The same concept, if you put salt on, if you eat too much salt, it dehydrates you. You feel that your heart is palpitating more. If you took note, you will notice that that's going to happen. You feel dehydrated if you take too much salt. Either you eat fries. Exactly, very good. Does it over all the same way? Sorry, I got. Oh, sorry. Okay, you're welcome back. Now, it's the same concept. So the issue about coffee, you feel the same way with coffee as well when you are not hydrated. All right. But what salt does to you is the same thing. I've noticed that it's the same thing. When you are not hydrated, obviously you should not drink coffee. See, ideal way, coffee is not the first thing you should drink in the morning. It's not healthy for the system at all. It's not healthy. It's not the first thing you should drink. Drink water first, then you can drink coffee. All right. But when you take, let's say you eat fries and you have not eaten, is this what is the struggle that is going to happen to the cell if you are not well hydrated? Because your cell is going to be dehydrated by that salt. Now, yeah, that's what happened there. I was going to use an illustration, but since it's going to be a recording, it may not be too appropriate. All right, let's go. I have time. Okay. Now, this is facilitated diffusion. Now, from the word facilitated, it means it is normal, regular diffusion that does not need energy. It is passive, but it needs to be facilitated. And if it needs to be facilitated, something has to facilitate it. The facilitator is a protein. All right. So again, no energy is required because it's diffusion. It's going to be moving from where it is large to where it is low. So now look at this now. These here are glucose molecules. In the extracellular space, there are a lot of them here. They are not existing in the cytoplasm. If you need to move some of them into the cytoplasm, glucose cannot move by themselves. They need help to be able to move. They cannot move. They cannot diffuse through the cell membrane. So they need something to help them. And that's where a protein comes into place. So, but because they are moving from where it is high to where it is low, they still fulfill the condition for diffusion, because diffusion means it's coming from high to low. Energy is not required. But in this case, for you to be able to proceed, because it's a large substance, it needs a facilitator. And that is where the protein comes into place. So It doesn't need energy. The glucose molecule just bind here, and then it will be released into the cell. It will bind here to be released into the cell. When it is, so you see, it will bind here, then it closes so that it can deliver the one it has taken. It will open again, pick it, then deliver it. That is a protein doing the job as a facilitator. Energy is not required because it's going from where it is high to where it is low. All right, let's go. Now, when we say active transport now, active transport simply means it is active. It is active means that it requires energy. Now, if it requires energy, that means it is not going from where it is high to where it is low. It means it is going otherwise. So when reactions or molecules travel from where they are high, from where they are low to where they are high, it is active transport. So if you look at this now, this channel is transporting potassium into the cell from the extracellular space. But you can see we have just one potassium here, or let's say 3 as it were here, but we have a lot here. You see, we have a lot here. So while transporting from where it is high, low to where it is high, if that's going to happen, then energy will be required. And it will also require a protein. So energy is required, a protein is required. The protein that is used here is sodium potassium pump. This is the pump, is a protein. All right, so chemical moves from area of lesser concentration, you see that, to area of greater concentration. And now it is not moved along concentration gradient, it's moving against concentration gradient. Concentration gradient means if it is moving along concentration gradient, it's moving from high concentration to lower concentration. But in this case, it's moving from lower concentration to higher concentration, so it's against. All right, movement of glucose. And again, you see here, movement of ions. This is what we mean by charged molecule, which will be cation or anion, as the case may be, right? Good. So that's what we call active transport. Again, look at this. This is, now the way this works is this. This is active transport. And the way it works is sodium is the major thing that this guy carries. Now, but anytime he's carrying sodium, that process generates a kind of energy that makes it to be able to take glucose with it. So sodium is the major guy that is being created, but anytime it's carrying sodium, it also generates energy to carry glucose on the long run. So sodium moves in, then glucose also moves in. Let's go. Now, the third kind of transportation, we talk about the passive, the active, and the third one is the vesicular transport. Vesicular transport simply means it requires vesicle, all right? It's not far from what we've been talking about, but this is a kind that requires vesicle for the substance to be able to get into the cell, meaning that a membrane sac will be created. It's more like you created a cloth, a jacket for the substance. So the substance is going to be in the vesicle, and then it's now. So this is what we are talking about. You see, these are ligands, substance that needs to get in the cell. You see the receptors, then they are going to penetrate the cell membrane, and then the cell membrane is going to create a vesicle for it. Now, this is the vesicle that was made. It is now inside the cell. So the substance will not come into the cell by itself. No. It will need to steal a part of the cell membrane to form a membrane, which is what we call vesicle for the cell, for the substance. So now it is inside the cell membrane, inside the cell. For you to get what is out, what is inside. So this is like a cell eating up large substances. And we call that process, when anything is coming into the cell, we call it endocytosis. All right? When it a substance is moved into the cell with the use of vesicle, endocytosis. When a substance is leaving the cell with the use of vesicle, exocytosis. So exo means exit. Endo means entrance into the cell. All right, so endocytosis means that a vesicle carries some substance into the cell. Exocytosis means a vesicle carries some substance outside of the cell. So what we are looking at now on this slide is a case of endocytosis. Now, there are three kinds of endocytosis. Endocytosis, a cell can eat solid material. That is what we call phagocytosis. A cell can also drink, pinocytosis. So you see, cell eating, cell drinking. And the third category is that the endocytosis could be based on receptor. It simply means the cell is selective. Because there's a receptor, just like this now, there are receptors on the cell that determines what the cell picks. This cell will only pick the things that can bind to this receptor. All right. Now, vesicle means it's from vesicle. So how will the ligands inside this vesicle, how will they come out? The only way they can come out is that lysosome is going to bind to the vesicle, and then it will break down the wall of the vesicle so that the vesicle can release what is inside it. That is what we call phagocytosis. If it is liquid, it's just the same order except that the cell is drinking, right? If it is receptor mediated, it's also the same thing we pictured here. So again, don't forget, anything coming into the cell through vesicle, endocytosis. Endocytosis can be three types. Phago, self-feeding on large substances, solid. Drinking, pinocytosis, and it can be receptor-mediated endocytosis. Then exocytosis means that the cell is releasing something out. Now, I will let you study these parts, the characteristics of each organelle yourself. Cytoskeleton. Key thing about this is cytoskeleton. There are three types of them. All right. There's microfilament, which is the smallest. So you want to take note of that, is the smallest. This is intermediate size, and these microtubules are the largest. So these are the largest. So you want to take note of that, largest and smallest. And this is middle. All right. Now, I will let you study the organelles. Please study the organelles by yourself. If I see anything that I need to mention particularly, I will. Yeah, study the organelles. Study the kind of ribosome we have. We have just one type of ribosomes. However, their location determines their classification. So the group one are the free ones. This one, you just see them in the cytoplasm. Let me show you a slide. Now, these are ribosomes. These small colors. These are ribosomes. These are ribosomes. These are ribosomes. All of these are ribosomes. These guys are what we call free ribosomes or unbound ribosomes. And here are ribosomes too. On the surface of this membrane, endoplasmic reticulum, which we call rough endoplasmic reticulum, they are ribosomes. So these ribosomes, we call them band of Feast. All right, so let's get back. And there are also some ribosomes on the nuclear envelope too. You see some ribosomes here. See some here. You see some on this membrane. Those are ribosomes. So ribosomes can be free or bound. The free ones, they are not connected to anything. They are just in the cytoplasm. The bound ones, they are connected to the cytoplasm or to the they are connected to the, not cytoplasm, they are connected to the rough endoplasmic reticulum or to the nuclear envelope. So that's what you have here for ribosomes. You see, they are scattered in the cytoplasm. Those are the free. Scattered in the cytoplasm, free. Band attached to endoplasmic reticulum or nuclear membrane. Those are band ribosomes. All right, let's go. What ribosome does anytime is to create protein, right? Yeah. Smooth ER, it is smooth because there's no ribosome. That's why it's smooth. This is rough because there's ribosome on it. The smooth also makes smooth substances, which is lipid, and it also helps with detoxification of your system. The rough makes makes protein. All right, Golgi apparatus. Majorly, what I want you to take note particularly of Golgi apparatus is that it does modification and packaging. So now let me show you what happens. I will go on this on the cell slide to show you what happens there. Look at this. Now, here is Golgi apparatus. Here's Golgi apparatus. Here is this rough endoplasmic reticulum. Here is the nuclear envelope. So ribosomes are here, and there are some ribosomes also that are free. Now listen, the ribosomes that are free, they make proteins. The proteins they make is for the cell itself. So I will write it there. Free ribosome makes protein for the purpose of the home cell. The home cell, the cell that makes that protein. Now, plant ribosomes, they usually make proteins that are for transportation. So these ribosomes now, when they make their protein, the protein is still raw, it's not 100% ready to use. So they will make their protein, and then the protein will come here, it will be packaged in vesicle. So the endoplasmic reticulum will also create a vesicle, just the same way we said the cell membrane created vesicle the other time, right? All membranes are able to create vesicles. And the reason they create vesicles is so that they can package their product in it for transportation. So endoplasmic reticulum creates vesicle. The vesicle travel towards the Golgi apparatus, like you can see here. Now, this vesicle will release the protein, the pre-proteins or the immature protein that has been made. Then those immature protein is going to travel through the disc. Each disc, you can see a lot of disc there for the Golgi apparatus. So it will pass through each of the discs for modification and for packaging. Then once it's done, it will be packaged. You see, it will be packaged again into vesicle. Then the vesicle goes to the cell membrane. You see that? The vesicle becomes part of the cell membrane, and then it will go out, package into vesicle, then travel to the cell that is going to use it. So that is actually what happened. What I just showed you now is what we call endomembrane system. It includes the nuclear envelope, the Golgi apparatus, the rough endoplasmic reticulum, lysosome, nuclear envelope, and vesicle. And cell membrane and the vesicles. Let's go. So I've talked about Golgi apparatus. Majorly take note of the functions that they do. Lysosome. Obviously, lysosomes have digestive enzymes. We call it hydrolytic. Hydrolytic means enzyme that is able to break down. You remember hydrolysis reaction, hydrolytic reaction. All right? Majorly, they digest foreign substances that are foreign to the body. You have bacteria, anything like that. White blood cells package them, the lysosome break them into pieces. Autophagy is what I think somebody had talked about cell rejuvenation sometimes earlier today. Autophagy is when the body looks at itself, assess itself, and feel like, okay, this structure is already getting worn out. If we leave it there, it's going to cause a lot of chaos for us. It's like somebody is in a working environment. In fact, this person is now old, maybe like 80, and they felt like, okay, you can't, even though you still like to work, we can't let you continue to work the way you want to work again, because you are going to be causing some mistake, making some mistake, and some few things will be a problem. And so they decide to say, let's retire this person. That's the same thing that autophagy is. And it's the ribosome that does that job. So autophagy is when your body look at itself and say, I need to clean up. Some substances have been here for a long time. they are not functioning at their best again, and it will be safe for the body for us to shut it down. So what the cell does is to say, okay, let's clean up these cells. And so they can recycle those cells, recycle those chromosomes, recycle those nucleic acid to make new ones. All right, let's go. Now, peroxisome. The difference between peroxisome and lysosome is that peroxisome contain what we call catalase. It contains catalase. And you know what this catalase does? Catalase helps to break down H2O2 and it will form H2O plus O2. Now, all right, Fidelia, I got it. Now, H2O2 will be broken down to form O2 and H2O. Oxygen and water is not toxic, but hydrogen peroxide is toxic to your system. So catalase breaks them down to make us safe. So we say that it oxidizes toxic substance, it helps to detoxify your system, and that's the major thing. So, but when you look at it in terms of structure, it's the same thing. In terms of structure, it's just like the shape of lysosome, except that it has catalase in it. All right. Mitochondria have two membranes, and that is actually where cellular respiration takes place in our body. Now, key thing about mitochondria is that there used to be this thought that mitochondria used to be a prokaryotic cell, a small cell by itself. And the reason there's that claim is that it looks so much like bacteria. Because when you look at mitochondria, it has its own DNA, and it's circular DNA just the way you see it in bacteria. It has ribosome, makes its protein synthesis by itself. It has double membrane. So that makes it look like what you see in bacteria. And now there's a thought that mitochondria and chloroplast, that they used to be small molecules by themselves, that were taken up by another cell through phagocytosis. You remember? Through phagocytosis here. All right? Taken up by another cell. So the thought is that mitochondria and chloroplast used to be there by themselves. They used to be smaller molecules that they were taken up by phagocytosis. And now they are part of another cell, which makes that cell to become eukaryotic cell. Which makes the cell to become either plant cell if it's chloroplast, or animal cell if it's just mitochondria alone. Yeah, so that was the thought, that was that idea. All right, mitochondria, cell function, three major functions that your cell does: cell metabolism, protein synthesis, cell division. Let's go. Now, metabolism, you remember we talked about catabolism, anabolism earlier when I talked about exergonic reaction and endergonic reaction, so I'm not going to talk about it again. And aerobic respiration means it requires oxygen. Anaerobic, so it requires oxygen for the breakdown of glucose. And so this is what you see in cellular respiration, which takes place in the mitochondria. And maybe part of cytoplasm, but majorly in the mitochondria. It's just like out of these 32 to 30, 36 to 38 ADP, it's just 2 that is made here. Just 2 of it is made here. The other 30 are made in this mitochondria. All right? Anaerobic means it does not require oxygen, and that is what happened in the cytoplasm. So what happened in the cytoplasm does not require oxygen. It is in the mitochondria you have oxygen. All right, so you see 2 to 4 ATPs are made. It's actually 2 ATPs that are made on the average. Yeah, because it's like you invest 2 and you get 4. So that means your profit is 2, right? Okay, let's go. Now, transcription, translation. I'll do this real quick, and that's it. We are done. Now, translation and transcription. Look at this on the screen. DNA I'd written this today. I'm just showing you again. So RNA, and usually this is mRNA because there are different types of RNA, right? Gives you, then proteins. Now, DNA is located in the nucleus. And so to convert DNA to mRNA, it also happens inside the nucleus. So process of copying DNA to make RNA, this process is what we call transcription. So this arrow is saying, oops, I'm so sorry. So DNA to make RNA is transcription. To make mRNA is transcription. All right. So DNA to make RNA is transcription. It happened inside the nucleus because the DNA is located inside the nucleus. Now, so process of copying DNA to make RNA, it requires this enzyme, which we call RNA polymerase, and it takes place in the nucleus. Now, after the DNA, you can see that is what you have here. This is DNA double strand. This is RNA single strand. After the DNA has been converted to RNA, protein synthesis cannot happen in the nucleus. Why? Why do you think protein synthesis cannot happen in the nucleus? So I'm waiting for a response now. Why do you think protein synthesis cannot happen in the nucleus? I'm waiting. I know you guys are tired now, and I'm rounding off soon. But why do you think that protein synthesis cannot happen in the nucleus?
Because the ribosomes are outside of the nucleus. It's in the outer area, in the spaces. That's where all the protein can be synthesized.
That's great. That's it. Why? So Merides, the question is, why can protein synthesis not happen inside the nucleus? It cannot, because ribosome is not in the nucleus. And ribosome is not going to travel into the nucleus to make protein. No. It is the mRNA that has been made here that has to travel. You can see it will travel out of the nucleus into the cytoplasm. In the cytoplasm, we have RNA, we have ribosomes. We have it right on the nuclear membrane. We have it on the endoplasmic reticulum, and we have it inside the cytoplasm. All right? So the process of converting mRNA to protein is what we call translation. All right? And ribosome is the one that makes protein anyway. Ribosome is just like a reader. It's like you wrote something for me in Spanish, and I cannot read Spanish. Then I got a reader to read it for me in English. So ribosome is simply reading mRNA in another language. So the difference between RNA and protein is different language. It's just a language. It's not anything. So mRNA will be read in a different language by ribosome, and that is what we call protein. So somebody sent me a letter, and I got an app that reads it for me in English because the letter is in Spanish. That app is ribosome. The English language that I'm getting is protein. The initial letter in Spanish is the mRNA. So ribosome is simply a reader, right? Now, there are 3 RNAs that we have. We have the tRNA, Number 2, we have the mRNA. And number 3, we have the rRNA. Now, the rRNA is what produces ribosome. The rRNA itself is produced by nucleolus inside the nucleus, and it will later produce ribosome. The mRNA is the message you are getting from DNA. So this is DNA, this is mRNA. The tRNA is a guide that helps to check mark the process. The tRNA.
Oh yeah, give me that one more again.
Do it again.
Yeah.
Request again.
Yeah, the tRNA.
Okay. The tRNA is the one that helps to check the activities of ribosome. So now, this is actually what I'm saying.
The professor going over your quiz.
What did you say?
It's like saying the professor's going over your quiz, checking your grades, something like that.
I didn't get that, Troy. Can you say it again?
Like, say you took a quiz. The tRNA is like the professor checking your grades.
Thank you. Now, let me say it now the way it is. When we want to make protein, what is the source of the protein? I'm going to do this on the screen. Look at this. Again, I've done it twice that the annotation kept cleaning it for me. So look at this. Right. Then the last one is protein. This protein that you have here is simply an expressive version of the DNA. Does that make sense to you?
Yes.
The protein you have here. So this is it. Troy.
Yes, I'm here.
My skin is dark. What's your skin color?
Light.
Now, the reason your skin color is light is because the gene that control your skin color is converted to protein. Let's say that my parents, let's say my father is white and my mother is black. All right. I'm going to get 2 genes for skin color. I'll get one from my father, one from my mother. Does that make sense?
Yes.
So in quotes, I'm going to have 2 DNAs in quotes. I'm going to have 2 genes actually, a gene for light skin, a gene for dark skin. Now, by the time the process happens in my body, the gene that is converted to protein is the gene that will determine my skin color. So if it is the black skin gene that is converted to protein, I'm going to be dark skin. If it is the gene for light skin that is converted to protein, I'm going to be light skin. The gene that what you see outside is a product of protein. Does that make sense?
So then that means there's no dominant genes. It's just whichever one it chooses.
Thank you. Now, the one that is chosen is the one we call dominant gene. The one that, so when you say dominant gene, is because it's expressed. And that means is that is the DNA that is producing the protein that is causing your physical appearance.
Well, there are no stronger genes. It's just the way that the body cycles it in. It's just, so all that expression is, oh, I got more dominant genes than you and this and that. That's all hocus pocus.
Yeah. So the reason we call it dominant gene is because that gene is the one that makes this protein. You know what we call this process? This process here? We call it gene expression. So meaning that this is a gene. The gene that is converted to protein is the one that is expressed. And the one that is expressed is the dominant gene. So now, Troy, let's say that
Yes.
your mommy is black and your daddy is white, and you end up to become light skin. Inside of you is going to be gene for dark skin. But it is recessive, it is not dominant.
Right.
When you are going to give birth to a child, even though you marry a light-skinned wife, your child may end up becoming dark skin because the recessive gene inside of you may end up being dominant in your child.
Wow, so that makes a lot of sense when you have different colored parents having different colored babies.
Thank you. Now, it's possible that both parents may even be white and their child may be black. Do you know why? Because if one of the grandparents had that skin gene that was given to any of the parents, but is recessive in the parents.
So it just lies dormant until it's ready to be used, basically.
Exactly. Now, it depends on what is being produced. What is producing protein is the one that is dominant. The dominant gene produces protein. Protein is what determines your physical. Protein is what determines anything that happens in your body. Your physical expression, your physical everything, your metabolism is determined by protein. And that's why people that don't that have lactose intolerance, for instance, is because they don't have protein for lactase. I mean, that should break down the lactose rather. So it is what anything that happens in your body is a function of protein. Lactase is a protein. All right, now so let's go. So the question you had asked me earlier, which is the reason I explained this, is tRNA, what tRNA does. So what you produce from DNA, oh, I should be rounding off now. What you produce from DNA is mRNA. That mRNA, okay, that's why I'm running off now as well. If you are here, just put here on the, let me see. Let me quickly round this off, then I will stop. DNA makes mRNA, mRNA makes protein. And the protein is a function of the mRNA. Now, so mRNA is what determines the protein. tRNA is going to always be opposite. tRNA and mRNA are like key and lock. That's why I say they help to check each other. I'll stop there. I can make more clarification on it later, but because of time for people that have to go and I need to take attendance. All right. So please finish the reading. And then we'll take it from there next week.